Okay, so I'm gonna talk to you about ketones and their roles on inflammation and inflammation resolution. I will define all these terms shortly, momentarily, but let's do a little bit of biochemistry since I'm the guy between you and your coffee and the guy between you and your bedtime in Europe. So I guess this is a slide which a lot of you have seen, but ketones, as you know, are products of fatty acid oxidation. Realistically, there's only, we call them ketone bodies, but chemically, there's actually one ketone. Anyone know which one that is? Look at the structure. Acetone, exactly. The rest are not really ketones, chemically speaking. So chemistry 101. Yep, they're still called ketone bodies. I mean, this is a carboxylic acid. Yeah. So you have BHB, beta hydroxybutyrate, acetoacetate, and acetone. Let's see if I can get the pointer to work. Yeah. That's fine.
Right click.
That's fine. I'll just wait. Oh, here. Okay. Yeah, got it. Okay. So basically, when you metabolize triglycerides, fatty acids get released. They go into beta oxidation. When that pathway gets saturated, you start producing ketone bodies. So here I'm showing the same pathway to the left, so that's what you saw in the previous slide. But I just also want to highlight a couple of things. Ketone body utilization. So a lot of you know they can be utilized for energy. So it goes into the Krebs cycle, and the product of energy is ATP, right? But the other thing that's maybe a little bit less appreciated, maybe Stephen, myself, and a few handful of others in the world appreciate it, is that ketone bodies can also be used for de novo lipid synthesis. So they also go into the malevolent pathway for cholesterol synthesis, and the malonyl-CoA can be grabbed by fatty acid synthase to make fatty acids. So now and this is critical for development and even maybe in aging, when you're trying to keep the structure of the brain or grow the brain in early life. And then there's another utilization or effect of these ketones, which is something that this guy, Junaid, discovered, I don't know, over a decade ago with his poor student, Taha.
Yeah.
But ketones, the ketogenic diet, also medium chain triglycerides, have this amazing metabolic effect in that they mobilize fatty acids from adipose to the brain. So this was one of the first studies to show that in rats fed the high ketogenic diet, brain DHA, or hexanoic acid, goes up, arachidonic goes up, all these polyunsaturated fatty acids increase in the brain. And in this study, we actually did use a tracer to confirm that, to track the mobilization. So you may want to think of ketones not just in terms of their energy effects, but also in terms of their structural effects for de novo lipid synthesis and or mobilization from adipose to the brain. So that's the metabolic effects of the ketogenic diet, low carbohydrate diets, of course, medium chain triglycerides. There's many ways, or prolonged fasting / starvation. So there's many ways to achieve the same outcomes. Another more direct way, as you've heard a lot today, is you can just give ketone salts or ketone esters and bypass all these metabolic effects. And in fact, in some cases, as we've heard and I've learned today, you may have different metabolic effects, whereby the ketogenic diet, for example, mobilizes free fatty acids from adipose to the brain, but ketone esters or ketone salts suppress free fatty acid lipolysis. So there's something different going on. But all increased beta hydroxybutyrate and acetone acetoacetate levels in the blood. Well, at least for these, you would increase acetoacetate and beta hydroxybutyrate. Now, it's well established that ketogenic diets have been shown to improve learning and memory in rodent models, of course. So this is a paper actually from UC, John Ramsey's lab, that was published recently, showing that with aging, if you give animals intermittent ketogenic diet, so I guess that sort of mimics intermittent fasting in a way. So you're taking the ketogenic diet on and off, or consistent ketogenic diet. you get you improve learning and memory. I'm going to stop scrolling first. On the Barnes maze and the Y maze. Also, in terms of, so this is age-related cognitive decline for Alzheimer's, and there's a lot of, this is one of many papers in the field, but this one is from my former boss, Mark Madsen at NIA. He was the chief of our section. So at the time when I was there, he published this study showing that ketone esters now prevent cognitive impairments in a triple transgenic mouse model of Alzheimer's disease. So learning and memory, and you have Mark listed all the stats here. Didn't show them on the figures, but I think the figures justify what is being said here. And then again, back to Cunane et al. In humans, the findings hold up. So when you give humans medium chain triglycerides, roughly 30 grams per day for 6 months, you do improve short-term memory on the free recall, verbal fluency, and on the Boston test. And importantly, a lot of these cognitive measures of cognitive improvement measures are related to blood ketone levels, so plasma beta hydroxybutyrate levels. So I think part of the question then is how are these things working, whether it's the ketogenic diet or the ketone esters. And part of the mechanism, we've discussed a lot the energy question, but there's also the anti-inflammatory hypothesis, and that has been brought up, I think, by you, Alex, right? So, and ketones have been shown to have anti-inflammatory effects, and this is another paper that just came out, well, a year ago, from UC Davis, Liu Wei Jin's lab, showing that if you add beta ABO, is beta amyloid on human-derived microglia, and you add beta hydroxybutyrate with the beta amyloid, you prevent markers of inflammation. So IL-1 beta, TNF alpha, both at the transcriptional level and the protein level. And this work is supported by other in vivo studies. So in models of spinal cord injury, other models of Alzheimer's disease, transgenic models. So you do have an anti-inflammatory effect of these ketone buddies, presumably whether produced by ketone esters, ketone salts, or ketogenic diets. So again, it brings us to the mechanisms. So is it, and I always think when we're dealing with dementia and Alzheimer's and MCI even, I don't think there's one thing happening at the time. In fact, if you want to target them, I think the system benefits from a multi-targeted approach because it's a very complicated disease and it requires a complicated solution. So we can think of the brain energy rescue as one mechanism. Brain structural changes shouldn't be disregarded, and this goes back to the de novo lipogenesis or mobilization of fatty acids that the brain desires, DHA, arachidonic acid, from adipose all the way to the brain. But also the question of inflammation. So when you're talking about inflammation, you can't think of inflammation without considering inflammation resolution. Now, these are two different concepts. A lot of people conflate anti-inflammatory diets with resolution. That's different. So I'll explain why. And resolution is really a concept that was brought forward by Charlie Sirhan at Harvard. So the idea is that if you have an inflammatory stimulus, virus, bacteria, air pollution, you name it, at some point, you're going to have a needed inflammatory response. You need inflammation to fight off pathogens, viruses, toxins. But at some point, you're going to enter a resolution phase. So the point of this inflammatory response is to sort of attack these invaders into your system. And then in doing so, obviously, there's cells that die, your own cells that end up dying. So resolution takes care of that. It cleans up the mess and promotes cellular repair and takes you back to homeostasis. And in a lot of diseases, including brain disorders, there's this idea that resolution never gets back to normal. So you end up with a state of chronic inflammation, right? So if you never resolve inflammation, then you're just going to be inflamed the whole time, right? And I'll show you some evidence to back that up, basically. But the idea is that is two phases under normal conditions. In pathological conditions, this goes astray. And the other point I want to make here is that, believe it or not, inflammation and resolution of inflammation is actually mediated by lipids, lipid mediators. That's what they call them. A lot of people think it's the cytokines, so the cytokines do play a role, but guess who the cytokines are coupled to? It's these signaling lipid molecules, previously known as lipid autocoids, hormones. Actually, over 30 years ago, they used to call them hormones because they're really potent at very low concentration. And these, chemically speaking, they're products of polyunsaturated fatty acid oxidation, both enzymatic and non-enzymatic. So let's also dive a little bit deeper into the terminology. So in chemistry, and I teach this to my students, they're rancid fats, right? It's oxidized fats, oxidized fatty acids. Food industry likes to call them primary oxidation products. And so when they measure shelf life of a food product, they actually measure these things and they call them primary oxidation products. Biologists call them many fancy names, and this is why it's so hard to sort of dive into this field, because what do you PubMed? Do you PubMed lipid mediators? Do you PubMed SPMs? If you PubMed SPMs, I guess you'll get a reference to Amazon because they sell SPMs. You guys know what SPMs are? Anyone?
Statistical mapping.
Close. Specialized pro-resolving mediators sold on Amazon for $40 American. So, yeah. So they sell
Use in the lab or to take? Yeah.
to take as a supplement. Yeah. So, our lab and others, we found that they're in fish oil. So now you can sort of fractionate the fish oil and you can sell them as SPMs. The question is absorption, and that's a different story. But yeah, so they're called SPMs. Another name for them is bioactive, so lipid autocoids, oxylipins. I'm sure you've seen this word before. And then there's oxylipids, and I still don't know what the difference is. I think oxylipins are thought to be, if you have an oxidized fatty acid that has a biological effect, it's an oxylipin. If you don't have a clean biological effect, it's an oxylipid. So it's like a useless oxidized fatty acid. So until you find an biological effect, it's an oxylipid. And of course, you know, some of the other terminologies, leukotrienes, eicosanoids, and resolvents. And I'm going to show you a nice picture that really highlights the fact that these are simply fatty acids with an oxygen attached to them. Nothing more, nothing less. I think food industry has it the simplest. They're rancid fats, and that's exactly what they are. In the bodies, we have a lot of enzymes that make these things because they're so important for signaling, right? So we have cyclooxygenase, lipoxygenase, cytochrome P450 enzyme, soluble epoxide hydrolase, and they can also be made by autooxidation. Although in our hands, when we do kinetics, it's really hard to oxidize these things by autooxidation. So hence, I guess nature invented enzymes. All right, let's also, naming things matters. The enzymatic products includes epoxides of arachidonic acid, AA, known as EATs or EPETers. So even the abbreviation, there's no consistency. You can Google EATs or EPETers or PubMed, and you'll have so many papers that use either or. I mean, what it stands for is epoxy, eicosa, 20 carbon, tri-enoic, so 3 double bonds, acids, right? I'll get to the structure in a second. And then you have metabolites of EPAs and EPEs. So these are epoxy, eicosa, tetraenoic acids, not tri. And then epoxy DPs. If anyone know what EPDP is? Yep, epoxy docosah pentaenoic acid. So 5 double bonds, 1 oxygen as an epoxide, and 20 carbons. So 22 carbons for docosah. So you need a bit of Latin help to get the names right. And then you have the hydroxides, which, so you'll have hydroxy, eicosa, benzenoic acid, etc. So the hydroxides of DHA in PubMed, they're called resolvents. Again, they're nothing more than rancid DHA, but they are called resolvents because in light of their role in resolving inflammation. So resolution. So that tells you something about their purpose. So would that be an oxylipid or an oxylipin now? Lipin. We have a purpose, so it's oxylipins. All right, you're not sleeping. That's good. And the last one is lipoxins. These are these are heat, so hydroxylated metabolites of arachidonic acid. That's one hydroxy group on arachidonic acid. And then it gets hydroxylated further into trihydroxylated molecules, which are called lipoxins. And then the resolvins are also trihydroxylated. So hydroxylations enzymatically with lipoxygenase yields a lot of these resolvins, also known as protectins, also known as neuroprotectin D1. So there's many names for the same molecule, lucky molecule. And then there's the non-enzymatic products, and that's it. So you've heard of the neuroprostanes, and then there's the ketones of linoleic acid and alpha-linolenic acid. Isoprostanes are of arachidonic acid. So isoprostanes, so these are non-enzymatically generated, and you do find them in the brain. The problem with these, I mean, not the problem, but there's not a lot of standards, analytical standards for them. I think there's probably one place where you can get them from, so it's hard to measure these most of the time. Well, you can screen for them, but we've never tried to measure them. So the other thing I want to point out that a lot of these lipid mediators have a short half-life. So this was actually one of the first tracer studies we did when I came to UC Davis. So don't worry about the top part, but just if you focus here on the half-life, this is 13 hydroxyoctadecadienoic acid. It's the metabolite of linoleic acid. It's an oxylipin because it has functional effects. It's pro-inflammatory. And we gave a deuterated tracer to rats, and we took blood samples over time, looked at tissues, and we can get half-lives. So I think just focus on this one here for the good vibe. But basically, what this tells you is that they're rapidly metabolized. So you're looking at a half-life in adipose tissue of less than a day. And I was actually quite surprised that adipose is so metabolically active compared to liver and compared to heart. But it is. So these things are coming in and out of adipose probably to maintain plasma levels. The plasma half-life is 1 minute in the rat. Same thing in the human. So these things are very low in concentration, and there's, we can get it. I can give a lecture on detectability and how do you qualify it. And the reason is they get into tissues. They also get esterified. That's some of our recent work, and they get turned over quite rapidly. So they're really they're fluxing all over the place. So they're bioactive, hence the name bioactive lipid mediator. So BLM is another abbreviation people like to use, BLMs. That's what it stands for. Because they're really involved in signaling blood flow, and some are sold as drugs. Can you believe it? The pharma industry sells you rancid fats as drugs. It's true. But this is carboprost, so it's used in youth labor, and it's a component of the abortion pill, right? And then this is alprostadil, PGE1, prostaglandin E1. It's actually used for treating congenital heart failure. It's a potent vasodilator. So these oxylipins are really high in bioactivity. And sometimes you can even win a Nobel Prize when you study them. So the last one was 30 years ago, but this was the Nobel Prize to Bergstrom, Samuelson, and Bain, who discovered the arachidonic acid eicosanoid pathway. Again, eicosanoids are a type of oxylipin. They're 20-carbon oxylipins, but they discovered that on the one hand, when you make these compounds, the leukotrienes, the prostaglandins, they discovered that aspirin targets this pathway, basically. So hence, the use of, this is the field of anti-inflammatory, when you take ibuprofen, aspirin, etc., right? So you're targeting these, you're really trying to target these bioactive lipid mediators, lowering prostaglandin E2 and other prostanoids. When you take baby aspirin, you're also trying to target the thromboxane for stroke, right? So I guess I think I've made my point that these are very bioactive people, right? Okay. This is what they look like, finally. What the hell did they look like, folks? Like I said, no surprise, like nothing, no big deal. It's oxygen on a fatty acid. So you have here the example of linoleic acid. Oh, my mouse is gone. It gets grabbed by lipoxygenase, it gets hydroperoxidized, and then it becomes the hydroxy linoleic acid metabolite or 13-hydroxyoctadecadienoic acid, and then this can become a ketone. So 13-octadecadienoic acid. On the other hand, it can become epoxidized, so you add an epoxy group to it. So this is epoxy octadecanoic acid, so one double bond, and then this can become a diol. These compounds, I think this one is called, I can't remember if it's the left or the right, but one of them was discovered by Bruce Hammack at UC Davis, 1997 Nature paper. At the time, he called it leukotoxin because it killed cells, basically, promoted apoptosis and killed cells. That's his 1997 Nature paper. And this one even killed cells more, the diol metabolite, so it was called leukotoxin diol. So that's another oxylipin. And as you can see, that if you take different substrates of fatty acids, you do get a lot of different biological effects. So from arachidonic acid, so what's shown here is that blue is pro-resolving, and red or pink, from my perspective here, it's pro-inflammatory. The magic here, the interesting thing is that some of them come from the same molecule. So a lot of times when you read papers, when you write papers, at least when I get papers to review, arachidonic acid is a precursor to pro-inflammatory lipid mediators. Right or wrong?
Right.
Yes and wrong.
Well, it is.
It is.
A precursor.
A precursor, right? But it's also a precursor to a lot of pro-resolving lipid mediators, right? So it's both. So when you say it's a precursor pro-inflammatory, that's half the story, because half of the arachidonic acid actually goes towards pro-resolving lipid mediators, specifically the epoxides, all of these EPs or EPeters here. And so these are potent pro-resolving molecules. DHA is easy, and the EPA are easy, so most of them go into pro-resolving lipid mediators. Actually, these should be. Sorry, blue is pro-resolving, red is pro-inflammatory. I said that, right? Okay, thank you. Linoleic acid, on the other hand, as I mentioned, goes into pro-inflammatory bioactive lipids. So I think it's complicated, right? So you can't just say, I'm going to give a fatty acid and I'm going to lead exclusively to pro-resolving, maybe except for DHA. But I'm sure there's some DHA metabolites we haven't discovered yet that are also pro-inflammatory. It's just a matter of time. And again, you grab them by the same lipoxygenate. You have LOX, lipoxygenate enzymes, cytochrome P450s for epoxidation, and cyclooxygenase enzymes for oxidation. Now, the reason why all this matters when you go to diseases like Alzheimer's disease is that actually there is truth to the idea that there's impaired resolution. So this is one of our recent papers. It's in Press, I guess. But where we showed, so on the left, let's start with the left, in a transgenic rat model of Alzheimer's disease. So this has develops both the beta amyloid plaques and the neurofibrillary tangles. So it has the beta and the tau pathologies. You get a wipeout, a 50% reduction in the concentration of pro-resolving lipid mediators in the brain. So this is transgenic. I'll get to the FA and TRAP in a second. And in this study, the novelty about it is that we're looking at risk factors for dementia, one of them being traffic-related or air pollution, which we're calling here TRAP. And these are rats exposed to actually car air pollution from the Bay Tunnel, California. And you can see that air pollution does the exact same thing as having the Alzheimer's transgenes compared to wild-type control rats. So what this is indicating is that if you're predisposed to Alzheimer's genetically, you do have a wipeout of your resolvins, pro-resolving lipid mediators, and air pollution does the exact same thing, which is probably why it disposes you to increased Alzheimer's. And in this study, it was previously published actually that these animals do have cognitive impairment. So they all look alike, except one has a transgene, and the other was exposed to air pollution. Does this matter to humans? And we're showing yes, it does. So to the right side is just, I mean, there's so many compounds in that paper, so I just chose my favorite, 1928 PDP. You can ask me why it's my favorite. But as you can see in controls with age, you sort of maintain your pro-resolving lipid pools in the prefrontal cortex. With Alzheimer's, with age, this sort of goes down, suggesting that in Alzheimer's, you do have an impairment, a reduction in pro-resolving lipid mediators and an impairment in resolution pathways. So I'm going to circle back to ketones shortly. And just to make the point, what do these things do? They actually do promote neuronal repair. So axonal outgrowth is a form of neuronal repair. So you can see that adding 1415 EAT, it's a metabolite of arachidonic acid, increases axonal outgrowth. When you add an inhibitor that degrades this EAT, this axonal outgrowth is further enhanced. When you give the two together, you get connected neurons magically. So. But this is just to tell you what these things are doing in the brain. They promote brain repair. Other studies have shown they promote neuronal survival. So they're doing a lot of things in terms of resolving inflammation. And that's what it's about. It's sort of getting you out of this damage, taking a damaged cell, repairing it, and keeping it. Because with neurodegeneration, you're losing cells. But here you want to keep them, repair them, and move on, right? And that's what these compounds do. So clearly, there's a need to develop strategies to decrease inflammation and promote resolution in neurodegenerative brain disorders, Alzheimer's being one of them. And so we hypothesized that based on the evidence showing that ketones are anti-inflammatory, so we thought that they would change the balance between pro-inflammatory and pro-resolving lipid mediators. Now, this is where the resolution question comes into play. So AIM-1, we tested the effects of a ketone ester supplement in aged mice. And then in AIM-2, we worked with Stephen. This is data that just came out 4 days ago, literally, from the mass spec. We looked at the effects of ketone salt supplementation in humans with Parkinson's disease and Alzheimer's disease. So let's look at, and the methodology we used what's called targeted ultra-high pressure liquid chromatography and mass spectrometry. Basically, it's targeted, meaning we know what we're looking for. If we don't know what we're looking for, we're out of luck. We're not going to find it. We just focus on some 80 compounds that we've created to capture a lot of these inflammatory and pro-resolving oxylipins or lipid mediators. So in the first study, we got aged mice. So 24 months old, and we put them on ketone ester diet. So this is a mistake. It's 20% of the calories, not 30%, relative to the control. So we replaced the ketone ester with carbohydrates, with cornstarch. So they were isocaloric, and we kept them on the diet for about 8 weeks. And we did some behavioral tests and took the brain for measuring oxylipins or lipid mediators. Behaviorally, I'm gonna just, so we did increase beta-hydroxybutyrate levels in blood. You wonder why, I think Philippe, mentioned in mice, the doses are so high for the ketone esters. And that's because we found many years ago that rats, mice, they have a problem maintaining ketosis. They're really good to load them up. If you do, you're good. If you don't, they just crash. Their ketone levels come down. So they're different. They're not human, and we're not them. So, but this effectively maintained ketosis. We looked at cognition, and we did a lot of tests, but this was on the Barnes maze. You can see clearly that the ketone ester group reduced the number of errors made in the Barnes test. And this effect was specific in females, but not the males. I haven't shown the male data because it wasn't significant. So it was a sex-specific effect. We then looked at the brain, looking at the inflammation resolution pathways. We didn't find anything in the pro-inflammatory lipid mediators. But what we did find is that there was, in a sex-dependent manner, in females, there was an increase. The ketone ester increased a lot of the pro-resolving epoxides of arachidonic acid, EPA, and DHA, including my favorite DHA lipid mediators, or SPM. So you can see that it increased it by some 50% in some cases, and this one was a trend, the one to your left at the bottom. it tended to be significant. And there's other examples, but I'm just showing you the gist of what's happening here. But basically, the ketone ester supplement did increase in the brain pro-resolving lipids, lipid mediators, or those involved in inflammation resolution. So obviously, I'm wondering the next study or the next grant I'm probably going to apply for is, can we use them to rescue the resolution deficit, the pro-resolving lipid mediator levels we saw in the transgenic Alzheimer's rats. So that's one potential use for these ketone esters. Summary for this study is that the effects were sexy. We found an improvement in cognition in females that corresponded to an increase in pro-resolving lipid mediators. And we're trying to see now if this is due to precursor fatty acid changes in the brain or structural changes, so myelin and other and phospholipids. Now we go to AM2, the study with Dr. Cunane. And this is when, please feel to correct me with the design, you and Valerie, but basically, this was a study a pilot study that explored the effects of the beta-hydroxybutyrate salt, so administered as beta-hydroxybutyrate 24 grams per day in patients with 10 patients with Parkinson's disease and 4 with Alzheimer's. So it was a before and after trial. 1 day of acute intervention here, where they did memory before and after. And then from after that, the acute phase, it was a 12-week it was a 3-month intervention. Look at my 2 months, looking at my French here, 2-month intervention, pardon me. Just like in the mice, looking at the effects of both exercise and the ketone ester supplement on memory. So what we did was we looked at the lipid mediators at baseline after the acute supplementation phase and after 8 weeks of exercise plus supplementation. And what we found, so I'll start with this compound, prostaglandin F2 alpha. It's a marker of, this is actually an auto-oxidation product. It's not enzymatically generated. But with the exception of this compound, there were no differences between the Alzheimer's and Parkinson's at baseline for the lipid mediators. And this one didn't change over time compared to baseline, but as you can see in the Alzheimer's folks, it's higher. And I think I'm not surprised because there's a paper by John Morrow and others showing that this compound is high in people in post-mortem brain of Alzheimer's disease patients. So it sort of validates our methods, and we're picking this up in the blood. Of course, we don't have controls here, but at least compared to Parkinson's, there's higher arachidonic acid-derived PGF2 alpha. Now, moving forward, we looked at the pro-inflammatory and pro-resolving lipid mediators. So what I'm going to show you now is the delta, the change in how these change between baseline and from 2 months to that 1 day after the acute supplementation. Oopsie. Okay. So, as you can see, again, if you express it as a line, you get the same answer, but this I just thought it was easier to show it this way. With acute supplementation, prostaglandin E2, which is pro-inflammatory, doesn't change compared to baseline, but after 2 months, it actually decreases. So the ketone ester decreased COX-derived prostaglandin E2 levels. And this is now in the combined cohort, Parkinson's and Alzheimer's, because there were no differences between the groups now. Looking further into the other bioactive lipids, the ketone salt supplement also reduced the LOX-derived pro-inflammatory lipid mediators. So you have the 9-HOTR, 9-hydroxyoctanoic acid, 13-HOTR, 9-heat, and this is actually 11-heat, pardon me. But it reduced all of these lipoxygenase-derived metabolites, which are mostly pro-inflammatory. It also decreased the sum of arachidonic acid epoxides, which are pro-resolving. It decreased them. And there's a good reason for that. I'll explain it in a second. But again, after 2 months, total AA epoxides decreased. In the acute phase, there were no changes. So what this shows is that the ketone salt supplement reduced pro-inflammatory lipid mediators derived from cyclooxygenase and lipoxygenase. What else targets cyclooxygenase, by the way? Aspirin, right? Ibuprofen. So it's sort of you're having a ibuprofen aspirin-like effect, right? Which is pretty cool. And the reason why it reduced pro-resolving epoxides, actually, this is in line with the recent rodent study we just finished, showing that if you suppress inflammation, you actually also dampen resolution. And the reason is because you don't need it, right? So you don't, if you don't activate, you don't need it. So in summary, last slide. In mice, ketones promote resolution of inflammation. In humans, they dampen inflammation. So resolution wasn't really activated because inflammation was dampened in the first place. Possibly because it's under control so far in these patients, although it would be nice to see how they progress over time. So I just want to thank my collaborators, of course, the students who did this, and NIH and the Alzheimer's Association for, there was a trickle of money that helped with the Parkinson's study. So thank you for your time and attention.